In the quest for new and effective medicines, scientists are increasingly turning to the intricate world of proteins. These complex molecules, the workhorses of our cells, hold the key to understanding diseases and developing targeted therapies. Structure-based drug design, a revolutionary approach in modern pharmacology, leverages the three-dimensional structures of proteins to design drugs with exceptional precision. Imagine a lock and key. By understanding the shape and properties of the lock, the protein, we can design the perfect key, the drug, to interact with it and elicit a desired therapeutic effect. This powerful technique has revolutionized drug discovery, allowing scientists to develop safer and more effective treatments for a wide range of diseases. By understanding the intricate dance between proteins and drug molecules, we can design drugs that bind specifically to their targets, minimizing side effects and maximizing therapeutic benefits. Structure-based drug design starts with unraveling the secrets held within protein structures. These structures, often determined using techniques like X-ray crystallography or cryo-electron microscopy, provide a detailed blueprint of the protein's architecture, revealing potential binding sites for drug molecules. Armed with this knowledge, scientists can embark on a journey of rational drug design, meticulously crafting molecules that fit snugly into these binding pockets. This targeted approach stands in stark contrast to traditional methods, which often relied on trial and error, screening vast libraries of compounds in the hope of finding a hit. The journey from a protein structure to a promising drug candidate is a multi-step process, each stage crucial in shaping the final outcome. It begins with preparing the protein structure, refining it to remove any errors or inconsistencies that might hamper subsequent analysis. This digital cleaning process ensures that the protein is in its optimal state for computational modeling. Next comes energy minimization, a computational technique that simulates the protein's natural movements and flexibility. Like finding the most comfortable position for a complex origami structure, this step ensures that the protein is in its most energetically favorable conformation. Once the protein is primed and ready, the search for potential drug candidates begins. Ligand generation comes into play, using sophisticated algorithms to design or select molecules that complement the protein's binding site. Imagine a sculptor meticulously carving a piece of clay to perfectly match a mold. This step involves finding or creating molecules that fit snugly into the protein's active site. Finally, molecular docking brings the protein and potential ligands together in a virtual dance. Like two puzzle pieces coming together, this computational technique simulates the binding interaction between the protein and ligand, predicting the strength and specificity of their interaction. By analyzing the results of molecular docking, scientists can rank order potential drug candidates, prioritizing those that exhibit the most favorable binding characteristics. Cheminformatics, a field that blends chemistry and computer science, provides powerful tools for navigating the vast chemical space in search of promising drug candidates. Imagine a vast library filled with millions of books. Cheminformatics provides the search engine and filters to help us find the exact information we need. One such tool is pharmacophore mapping, which identifies the essential features of a molecule that contribute to its biological activity. Like identifying the key ingredients in a recipe that give it its unique flavor, pharmacophore mapping helps us understand what makes a drug effective. Quantitative structure-activity relationship, or QSAR, studies take this a step further, building mathematical models that correlate molecular structure with biological activity. Imagine having a formula that predicts how changing the ingredients in a recipe will affect its taste. QSAR helps us understand how tweaking the structure of a molecule will impact its potency or toxicity. Similarly, quantitative structure-property relationship, or QSPR models, predict other important properties of a molecule, such as its solubility or metabolic stability. This information is crucial for optimizing drug candidates, ensuring they are not only effective but also easy to administer and process by the body. Screening different libraries of compounds is another important aspect of cheminformatics in drug discovery. Virtual screening allows us to quickly evaluate millions of compounds against our target protein, using computational power to prioritize those most likely to be effective. This significantly speeds up the drug discovery process, allowing us to focus on the most promising candidates. Discovering a promising drug candidate is only half the battle. Rigorous testing is essential to ensure its safety and efficacy in humans. Pharmacokinetic analysis plays a critical role in this process, evaluating how the body absorbs, distributes, metabolizes, and excretes the drug, known as ADU. Imagine tracking a package's journey from the warehouse to your doorstep. ADME analysis helps us understand how the drug moves through the body. Toxicity analysis is another crucial aspect, identifying any potential harmful effects of the drug. This involves a battery of tests, both in vitro using cells or tissues and in vivo using animal models to assess the drug's safety profile. Networking pharmacology offers a holistic view of drug action, mapping the intricate web of interactions between drugs, proteins, and biological pathways. This systems-level approach helps us understand the broader biological context of drug action, identifying potential side effects and uncovering new therapeutic applications. Gene ontology and gene enrichment analyses provide further insights into the biological processes affected by a drug. By analyzing the genes and proteins involved, we can gain a deeper understanding of the drug's mechanism of action and potential therapeutic benefits. Finally, network analysis helps us visualize and interpret the complex relationships between genes, proteins, and pathways involved in drug action. While the focus of this essay has been on drug discovery, these principles extend to other fields such as cosmeceuticals. Whether developing new powders, creams, dentifrices, lipsticks, or shampoos, understanding the interaction between molecules and biological systems is paramount. By harnessing the power of structure-based design and cheminformatics, we can create safer, more effective, and more personalized products across a wide range of applications.